Okay, we will make a start now. So welcome everyone to our A Level AQA Biology session. In today's lesson, we are going to be looking at the structure of prokaryotic cells. We're going to be looking at the structure of viruses. It's not a very big topic. It's only sort of a couple of slides, but our main focus is going to be exam practice for today's lesson. Last lesson, we looked at eukaryotic cells and the structure of eukaryotic cells. After we've looked at the structure of prokaryotic cells and viruses, I've selected a healthy amount of exam questions that we can look at. Okay, so if we begin now, this is what the specification says, and this is what we're going to be following for today's lesson. So we're going to be looking at the structure of prokaryotic cells. We're going to acknowledge that they're smaller than eukaryotic cells. We're going to have a look at these key points over here, and the differences that prokaryotic cells have compared to eukaryotic cells. So as you can see, prokaryotic cells have one or more plasmids, a capsule surrounding the cell, or one or more flagella. And then we're going to end off on this tiny part of the spec, which is all about viruses. Okay, at any point, if you have any questions, you can let me know, and I'll be more than happy to answer them for you. Okay, so we're going to start off by looking at prokaryotic cells, as I mentioned, and I only have sort of two slides on prokaryotic cells, and this is just about the structure of prokaryotic cells. So the first thing that you need to be aware of is that prokaryotic cells contain circular DNA. Okay, in eukaryotic cells, the DNA is much larger, it's linear. Okay, in prokaryotic cells, the DNA is circular. Okay, and we'll have a look at an image shortly. But the first point that you need to know is that prokaryotic cells don't have normal linear DNA. It's in a loop and it's circular DNA. They have a DNA, all the DNA in a loop, and they have circular DNA. Prokaryotic cells, as we just saw on the slide before on the specification, it states that they are smaller in size compared to eukaryotic cells. Eukaryotic cells have many subcellular organelles, and prokaryotic cells don't. They are much smaller in size than eukaryotic cells. They don't have a true nucleus or a nuclear envelope. Remember, the nuclear envelope we looked in our last lesson is literally the membrane which surrounds um, the nucleus, and it's the part where the nuclear pores are also present to allow mRNA to move into and out of the nucleus to allow the transcription and translation of proteins. So there's no nucleus or nuclear envelope in prokaryotic cells. And the key point, very important point that you need to know is that the cell wall is made out of murin. Okay, now murin is really important. Make sure you note this down because it always comes up again and again. Like sort of very often because the cell walls are made out of different um, sort of chemicals. In bacterial cells, they have a murin cell wall. Okay, so it's really important because that's what antibiotics target. They can target the sort of urine cell wall, the chemicals, they are attracted to that specific chemical. And that's why it's really important that you understand that the cell wall in prokaryotic cells, specifically in bacteria, is made out of murin. Okay. If we move on now, so these are the first four key differences that you need to know about the structure of prokaryotic cells. Contain circular DNA, they are smaller in size, they don't have a true nucleus, they only have a loop of DNA. If they don't have a true nucleus, they're not going to have a nuclear envelope. Therefore, they're not going to have any structures of the nucleus look like a nuclear pore. The cell wall is made out of murin, and murin is a glycoprotein. Again, we will look all on glycoproteins in a further unit. Now, the next few things that I'm going to touch upon is the cytoplasm. In prokaryotic cells, the cytoplasm contains enzymes and other soluble molecules. However, the cytoplasm, unlike in eukaryotic cells, it lacks in membrane-bound organelles. Okay, so for example, the mitochondria, chloroplasts, um, in terms of membrane-bound organelles, so smaller subcellular structures within the cell, it lacks in those subcellular structures because normally all subcellular structures, apart from ribosomes, will have a membrane surrounding them. Prokaryotic cells don't have that many subcellular molecules or subcellular organelles. Therefore, we say that they lack in these membrane-bound organelles. It's more of an empty cytoplasm with a few subcellular structures. Some also have the flagellum, and that's used for locomotion. It allows the bacteria, for example, to move and sort of transport from place to place. They also have plasmids, which possess genes which may aid the survival of bacteria in adverse conditions, produce enzymes which break down antibiotics. So that's the actual function of plasmids. And you've probably heard that in bacterial cells, you have plasmids. And now this is the function that you need to be aware of. Plasmids are contain specific genes and they help the bacteria to survive. And an example of how they can help the bacteria to survive is that they produce these enzymes which break down antibiotics, because ultimately antibiotics are there to get rid of the bacteria, they're there to kill the bacteria. And if the bacteria wants to survive, these plasmids will need to produce enzymes which break down antibiotics. And that's how you get resistant strains of bacteria. Um, if the type of protein that is being produced, if the enzymes which are being produced or being released by bacteria change. Therefore, they are able to break down the antibiotic more effectively than the previous bacteria were doing. And all of this happens when the bacteria adapt to the conditions that they are placed in. And then that bacteria can then go on to multiply by binary fission continuously until that bacteria can spread out everywhere, and suddenly you have a resistant strain of bacteria which is released into the environment, which everyone can contract. Okay, 70S ribosomes. We looked at 70S ribosomes in our last lesson as well. 70S ribosomes are the smaller versions of ribosomes. If you didn't already know. The other type that you have are the ATS ribosomes. Okay, and ATS ribosomes, these are present in eukaryotes. Okay, now that's because eukaryotes are much bigger. Now, 70S ribosomes, they're present in bacteria. However, they are also present in the mitochondria and chloroplasts as well. Okay, so they're also present in the mitochondria and the chloroplast. And that just gives you an idea of how the small bacteria or prokaryotes actually are, because these same 70S ribosomes are also present in subcellular organelles, which are present in eukaryotic cells, mitochondria, and chloroplasts. Okay, and finally you have the slime capsule. Okay, so a slime capsule protects the bacterium from other cells and helps the bacterium to stick together for further protection. You call it a process called clumping. Okay, so imagine one bacteria is that small. Okay, it's going to be even smaller than that, but one bacteria is that small. If this is fighting off a massive infection, if this is fighting off, we'll say an infection over here, that bacteria is going to be very useless or very slow by itself. And that's the purpose of the slime capsule. The slime capsule will help multiple bacteria literally come together and clump together to form a big, sort of a big bacteria like this. And this is called clumping via the slime capsule. And then this bacteria can go and fight off infections, as an example there. Okay, and if this is acting together as a team, they are also supporting and helping the bacterias or the bacterium which are inside this clump. So there are some bacteria which are shielded from the outside bacteria, and that's the advantage of having a slime capsule. They allow the bacteria to literally stick together for further protection of each of the bacteria. Okay, so those are the structures of prokaryotic cells, and that's the depth that you need to be aware of. Um, there are a few exam questions that we are going to be looking at, which surround the structure of bacterial cells. Um, But ultimately, even when we have a look at the spec, it says prokaryotic cells are much smaller than eukaryotic cells, which we have covered. They also differ from eukaryotic cells in the fact that cytoplasm lacks in membrane-bound organelles. We've looked at that already. They have smaller ribosomes. Remember, they have 70S ribosomes and not 80S ribosomes. Okay, they don't have these 80S ribosomes, they have 70S ribosomes. They don't have a nucleus, okay? Instead, they have single circular DNA molecule that is free in the cytoplasm and is not associated with proteins, okay? So if you guys remember, the DNA which is present in eukaryotic cells is histone bound, okay? So if I just add that in, we've not looked at it yet, but basically DNA in eukaryotes um, is bound to a protein which is called the histone protein. Okay, whereas in prokaryotes, the DNA is not bound with any proteins. In eukaryotes, you have the DNA which is tightly coiled because of the histone protein. The histone protein allows that tightly coiling of DNA. Okay, um, and prokaryotic cells have a cell wall that contains murin, which is a glycoprotein. Okay, again, really important point there. Murin cell wall is the type of cell wall that bacterial cells have. Now, prokaryotic cells also have additional features. They also have one or more plasmids, which you should remember from GCSE. They have a capsule which surrounds the cell, again, the slime capsule, which allows the clumping of many bacterial cells. And they have one or more flagella, which allows in locomotion. Okay. So that's what we've looked at. And literally it ends there. It says details of these structural differences are not required. So unlike the eukaryotic cells where you needed a bit of depth into the structure, that's not required for prokaryotic cells. Okay, and now what we're going to do, we're going to have a quick look at an example of a bacterial cell, and then we're going to move on to this final part of this topic spec by looking at viruses, and then we're going to move on to some exam questions. Okay, so prokaryotic cell. So here's the image that I've pulled up, and this is a bacterial cell. So as you can see, you have a cell surface membrane, which is again very normal. You know what a cell surface membrane is. You have a cell wall. You have this slime capsule, which is in the lighter green, and this allows attachment to other bacterial cells. You have these ribosomes. Again, these ribosomes are 70S ribosomes. They are smaller. Okay. The DNA, again, the DNA is in a loop and is not bound to a protein. Okay, i.e. it's not histone bound. It's just circular, it's in a loop, and it's not really doing anything. It's just there, it's not bound to a protein at all. You've got these plasmids which aid the bacteria in adverse conditions. And then you've got this flagellum, which is literally there for locomotion to allow the bacteria to move and carry out its function. Okay, so there's an example of a bacteria. Okay, and that's everything that I'm going to do on prokaryotic cells. What we're going to do now is move on to the structure of viruses. Now, there's only one slide on viruses. Again, it's not that sort of information heavy. It's just some key facts that you need to know and that you get assessed on. First of all, the point that I would like to bring up is that viruses are not classified as prokaryotes. And the reason why is that viruses are not classified as a living cell. They literally have very, very, very few subcellular organelles. And there are some properties of viruses that you are required to know. Starting off, viruses are acellular. If cellular means made out of cells, acellular means not made out of cells, okay, or divided into cells. Okay, so cellular is a cell, it's made out of cells. Acellular means that it's not made out of cells. Okay, so that's the structure of a virus. The first property is that it's acellular, it's non-living, um, and it's not made out of cells. Okay, you could remember this as being non-living. Okay, it's not made out of cells. The second thing is that it does contain genetic material, which can be DNA or RNA. Again, nothing sort of too difficult to remember. It's the same as prokaryotes, and it's the same as eukaryotes. It contains some genetic material because you obviously need to know what the virus needs to do. It also has a capsid, which is the protein coat of the virus. Okay, it's the protein shell of the virus enclosing its genetic material. So obviously the capsid will look something like this, like that. And inside you will have your DNA or RNA. Okay, and this is the protein coat of the virus, which is the capsid. Okay, so that's the protein coat of the virus. It is a protein coat, just so you know what it is. And it encloses the genetic material. It's where the genetic material sits. Okay. And then finally, you've got attachment proteins. Okay, so there are specific attachment proteins which are present on viruses. Okay, and they normally stick out like so, like that, but just all around the virus particle. And why is this? Well, they act as the key to unlock host cells by interacting with the lock on the cell surface. Okay, so imagine we say that viruses live inside cells. How do they actually get inside cells? How do they know that that's a cell? Well, they have these specific attachment proteins. Okay, so these are your attachment proteins. And these attachment proteins are basically what attach to the host cell. So normal body cells, the virus particle attaches to via these attachment proteins. And these attachment proteins are complementary in shape to the ones which the body cells normally have. And it will act as a key and lock. So it will act as um, a lock and key, if you remember the enzyme model, but these will literally attach to the receptors which are present on normal body cells. Um, these receptors are present on normal body cells. Okay, so the attachment proteins are also contained within a virus. They will attach to the receptors that are present on normal body cells to allow the virus to enter. Okay, and the reason why you don't want to be catching a virus is because the virus will live inside the cell. It will follow the biological processes which are inside the cell, and it will literally multiply very quickly. Okay, and the reason why you also don't want it is because that virus will have attachment proteins which are complementary in shape to the receptors on your normal body cells. And once it's inside the cell, it will keep multiplying until it will cause cell damage. Until all the contents are released, the cell bursts because it's holding too many virus particles, and that's when you feel ill, basically. Okay, so that's the structure of viruses. Key things, it's acellular, which means that it's not made out of any cells, it is non-living, um, contains genetic material, either DNA or RNA. How is the genetic material going to be protected? It will need something to be protected, and that's what the capsid does. The capsid is the protein shell of the virus enclosing its genetic material. And then finally, the virus also contains these attachment proteins, and the attachment proteins are basically the key or the specific shape which attaches to the receptors on normal body cells, allowing the virus to enter into the normal body cell and cause damage. Okay, so that's basically your structure of viruses. And if you go back to the spec just as a reference, over here, viruses are acellular and non-living. We've covered that again. The structure of virus particles include genetic material, capsid, and attachment protein. And because it doesn't say that the structural differences are not required, like it did with prokaryotic cells, you will need to know the structural differences. You will need to know the structural functions and anything like that, which we have also already covered. So as you can see, it's a hefty part of the spec. However, there's not much that we need to go and learn. It's pretty much sort of stuff that you will come across all through biology, and it's just about building upon those concepts. Okay. Um, so that is everything in terms of the content for the structure of prokaryotic cells and viruses.